This is my enzyme lab presentation, and an enzyme is a substance that's produced by a living organism that acts as a catalyst to bring about one specific chemical reaction. And they speed up chemical reactions by lowering the activation energy for the reactions, and the lower the activation reaction, the faster the speed of the reaction. And enzymes are important for all living organisms, and this is because they are needed for metabolic pathways in the body, respiration, digestion, and so many more things. And when enzymes function properly, homeostasis is maintained. This type of enzyme that we studied, trypsin, in the lab, breaks down polypeptide bonds of proteins into smaller bonds and then into amino acids. In humans, trypsin is found in the small intestines and helps digest food and other processes in the body by hydrolyzing proteins. Trypsin is also found in animals, although it can slightly differ in its functions. It can differ in its activity, whether carrying out digestive functions or regulatory functions. And as I mentioned before, its digestive roles includes breaking down large polypeptide bonds into smaller amino acids. And trypsin's regulatory functions is similar, but instead of carrying out that breakdown of certain polypeptides, it instead activates other proteins through proteolysis. Trypsin is secreted from bacteria, and therefore it has an optimal operating pH levels and temperature vary within them. Our question for this lab was, what is the optimum pH range for the highest enzyme activity? And what we hypothesized was that the highest enzyme activity will occur from a rate of 8 to 9. We thought this because during our time before our lab, we did some research and found out that in humans, the pH of trypsin is level 8. Therefore, we predicted that the reaction buffers with a range of 8 to 9 will yield the optimum activity level for enzymes. During this experiment, we collected tubes. Each of the tubes contained 450 microliters of the reaction buffer, and each conditioned tested experiment was pHs 7, 8, 9, and 10. And they each contain 0.15 molar NaCl, which was across the board for all the tubes. And then also we incubated them at 37 degrees Celsius. This was because that was temperature that's similar to the human body. We also added 50 microliters of trypsin, and after we incubated them for 20 minutes, at the 37 degrees Celsius, we added the TCA, trichloritic acid. That was the last component to be added to the tubes, and this was to stop the reaction from happening, so that once we got after we microcentrifuged the tubes, we would take the supernatant and transfer to a clean cuvette so that we were able to find an absorbance reading at 440 nanometers. The higher the reading of absorbance, the higher the rate of reaction. We also did negative controls where in each of the tubes we added water instead of the trypsin. We did this because we wanted to make sure that when there wasn't any trypsin, there was no reactions occurring outside of the enzyme's reaction with the azocasein that we added to the tubes, which we actually added 500 microliters to each of the tubes of azocasein, which was the protein that the trypsin was breaking down during the experiment. We also did 3 trials of each, so for each pH of 7, 8, 9, and 10, we did 3 trials for each of them. We did this to make sure that we got multiple results so that we could scale out large numbers, small numbers, error, anything like that. Make sure that we're getting the most accurate results for the experiment that we could get, so we could really tell which pH had the highest absorbance level, which had the highest rate of activity in the enzymes. We also, you can see in our results, when we conducted the t-test, we were able to see that none of our p-values were significantly different, which means that we can't use our data to support our hypothesis, which you can see that even from 7 and 8, 8 and 9, 9 and 10, none of them were significantly different. And we can see that the absorbance for pH 7 was approximately 0.38 with the error bars, and the 8 had an absorbance of 0.48. That shows that there's an 80% chance of similarity between the two pHs, and pH 9 has an absorbance of 0.45 as an average, and there's an 80% chance that these two pHs are also similar. So the variability among these trials is small at 5 to 10% average, and you can see that all of these numbers, it's not that far apart. Yes, you can say that pH of 8 had a high absorbance, more than all the other ones, but you can see that it's not that far off. And when the p-values that we calculated, our hypothesis was not supported because all of our p-values were greater than 0.05. The difference between 7 and 8 was our lowest p-value, closest to 0.05, but it was actually 0.72, which is still higher than 0.05. This means that the data recorded in the experiment is inaccurate. We can't use it to support our hypothesis, and we can't use it to say anything about what we predicted. We predicted that the highest enzyme activity would be pH range of 8 to 9, but we cannot support that because we don't have any evidence that could support that due to error. Therefore, it's just inaccurate and we cannot use it. Compared to groups C and D, in their experiments, there were groups who had the independent variable as pH as well. Their results all turned out to be conclusive, unlike ours, and it appears that there were some conflicts between the results. In group C, their results show that a range of pH 7 to 8 was optimal, whereas group E showed pH range of 8 to 9. So it's kind of interesting how different groups got different results and were able to support their hypotheses. But we can also think that maybe they had different temperatures, and that could have also affected the rate of reaction. We saw in groups before for the first experiment that groups who had a temperature at 37 degrees Celsius had higher absorbance, so we decided to go with that. But maybe other groups decided to stick with room temperature, and their absorbance was lower, which could have skewed some things, or there could have been error. Since our experimental design mirrored the other group's designs, their data was supported by their hypothesis, our experimental design is valid, but what we suspect the cause of our inclusive data is error and improper execution of the experiment. Some of the possibilities of this error could have been imprecise measurements when adding our reagents, or cross-contaminations between our sample, or different controls, experiment groups. We could not determine any specific error that we remember during the experiment, but these factors could contribute an error and variability in our data. Our choice of our control group variables conditions cannot result in the highest possible rate of enzyme activity because group A data shows that a temperature increase, enzyme activity increases. I think they had up to like 60 something degrees Celsius. I don't remember, but it was very high, and the enzyme activity just kept increasing as the temperature increased. We don't know where it stops or plummets because they didn't get that far, but we chose a temperature higher than 37 degrees Celsius, then maybe we could have gotten more absorbance than a highest one of 0.458 as an average for the pH of 8. While our data was inconclusive, our experimental design is an improvement from our first attempt. Some potentials improvement in design between the first and second attempt, we were able to have more experience in doing experiments like these. In executing the experiment, we tested a broader range of pH to sort of see if it plateaued or if it continued to rise. As we could see, it does decrease, but we can't make any assumptions about our data because it is inaccurate and we cannot use it. While our conclusions to the experiment are inconclusive, if we were to run the experiment again with better execution, it is possible to find the ideal pH range for trypsin to break down proteins. Yeah. Thank you for watching.